Well, the development was just accidentally. So in 2012, I got the idea that this small molecule, acetyl-L-leucine, which had been shown in animal studies to be effective on acute vertigo, acts via the cerebellum. And therefore I thought, why not using a molecule which acts via the cerebellum in cerebellar diseases? So then we did a first case series, patients with different types of ataxias, and we saw a clinically meaningful benefit. That was the beginning of the story. And later on, we switched to other diseases with the leading symptom of ataxia, and we have started with lysosomal diseases, namely so-called Niemann-Pick type C and gangliosidosis type 2 Tay-Sachs disease. And first we did a phase 2 study and later on a phase 3 study. The latter on NPC was published in the New England Journal of Medicine in 2024. And based on these studies, we have got approval by the FDA and the EMA for the treatment of Niemann-Pick type C. Further on, we have broadened our spectrum of diseases which can be potentially treated by this small molecule. And this was also based on preclinical animal studies at the Department of Pharmacology and Biochemistry in Oxford, Francis Platt and Anthony Gaglioni. So they use the appropriate animal models of NPC and GM2. And from these studies, we learned that one basic mechanism of this molecule is to improve glucose metabolism. That means per glucose molecule, more ATP is produced, which brings cells in a better shape. Other mechanisms of action are normalization of excitability of neurons and also primary improvement of lysosomal function, namely via the so-called transcription factor B, TFAP, which was done by Anthony Gaglioni. So in other words, this molecule has multiple mechanisms of action. Later on, we have performed a phase 3 trial in ataxia-telangiectasia. This is a disease which is based on an impaired function of a DNA repair gene. So at first I was a little bit skeptical. Do we see a benefit in a disease with a totally different mechanism? And luckily, the clinical effect was even stronger than in Niemann-Pick type C. And the paper was published now in the Lancet Neurology, end of June. And of course, one will try to get approval by the FDA and later on the EMA for this small molecule also in ataxia-telangiectasia. Another thing, of course, in any treatment is safety. And it has been shown that this small molecule, a modified amino acid, has an excellent safety profile. This was expected. What is so special about this molecule and the acetyl group? Well, due to the acetyl group, leucine is transported by so-called monocarboxylate transporters with a very high capacity. This was also measured by some studies in animals. In other words, if you take leucine, it does not show up in the brain in relevant concentrations. Due to the acetyl group, it appears in very high concentrations inside the cells. So this is very special about this small molecule. Leucine alone does not work. The acetyl group is important. And another detail, we have an L form and a D form. Originally, the racemate with the D and the L form was used. These days, we use the L form only because the L form is the bioactive component. This is also due to the fact that we do have transporters for the L form and not for the D form. So L amino acids are physiological, D amino acids are counterproductive. They even inhibit the transporter. What are now the perspectives? So the next trial, and the trial protocol is ready, will be a trial on so-called CACNA1A neurodevelopmental disease. This is a new ICD-10 term since October last year, and it covers a lot of different phenotypes. Most familiar to most neurologists is episodic ataxia type 2, familial hemiplegic migraine, but there's also a neurodevelopmental disease which leads to severe persisting dizziness, imbalance, and ataxia. And this is going to be the next target, the next clinical trial, and we expect to recruit the first patients in Q3, Q4 2026. We have also demonstrated that this molecule has a neuroprotective effect. And the paper was published in Neurology, showing in the extension phase of the phase 3 trial that we can delay the progression of the disease in patients with Niemann-Pick type C. In other words, after 24 months of treatment, the patients with NPC are in a better shape than before beginning of the treatment. And the difference between the treatment group and a natural history group was 120%. So to make that clear, the molecule has a symptomatic effect, which you see after about 6 to 12 weeks. And in addition, on top of it, it has a neuroprotective effect, which delays the progression of the disease. And this brings me to another paper, which we published in Nature Communications. We had patients with so-called prodromal Parkinson's disease. They have a so-called REM sleep behavior disorder. If you suffer from REM sleep behavior disorder, the risk that you're going to develop manifest Parkinson's disease with motor symptoms within the next 10 years is about 85%. And we had access to a cohort of these individuals with a REM sleep behavior disorder. And we also performed a DET SPECT to measure the function of dopaminergic transmission. And after 24 months of treatment with acetyl-L-leucine, there was an improvement of the dopamine transporter function, which is very exceptional. So this molecule has a symptomatic effect and a disease-modifying effect, which we demonstrated in NPC, which we demonstrated in GM2, but also in prodromal Parkinson's disease. This molecule, beginning as a small baby, has now grown up, is in its late puberty, and we expect many more positive findings in the near future.